Hi and good morning. My name is Najihah binti Shahrun with number matric A193943, and today I will be presenting on dysbiosis focused on obesity. Human gut is a highly complex community of microorganisms, mainly bacteria, but also viruses, fungi, and archaea. Interestingly, most of these microbes are non-pathogenic and live in a symbiotic relationship with us. They are predominantly located in the large intestine, where the most dominant phyla are Firmicutes and Bacteroidetes. This microbial community also plays a vital role in forming multidirectional connection with other organs. The gut organ exists. Even Hippocrates, the father of medicine, said that seat in the bowel highlights how central the gut is to the human health. The gut microbiota, eubiosis, is important in metabolic function, in fermenting complex carbohydrate into short-chain fatty acid like butyrate and acetate, essential for maintaining gut health and energy production, synthesize vitamins such as vitamin K, and helps metabolize drug and xenobiotics. They also regulate both innate and adaptive immunity, for example, stimulating cytokines against pathogens like Citrobacter-rodentium. The short-chain fatty acids produced maintain the structural integrity and aids as a barrier by secreting mucus and antimicrobial peptides. Gut microbiota also suppresses the colonization of pathogens, including Clostridium difficile, pathogen that causes diarrhea with biosynthesis of secondary bile acids. Gut microbiota communicate bidirectionally with the brain, regulate neurotransmitter, and even influence microglia development. Under normal condition or eubiosis, the gut microbiota support digestion, metabolism, immunity, and even heart and brain health. However, when disturbed through poor diet, stress, lifestyle, or medication, it will lead to dysbiosis. This state will be defined where reduced microbiota diversity, loss of beneficial microbes, or an increase in harmful microbes. Dysbiosis disturbs normal physiological and metabolic processes, causing high risk for chronic diseases such as cardiovascular disease, inflammatory bowel disease, as well as autoimmune. So how dysbiosis contribute to obesity? A 2025 meta-analysis revealed that obese individuals showed reduced microbiome diversity where Firmicutes increases and Bacteroidetes decreases. A high-fat diet also will weaken the integrity of the gut barrier. Firmicutes are efficient at fermenting indigestible polysaccharides. Thus, it will lead to increased production of short-chain fatty acids. Normally, short-chain fatty acids are beneficial in regulating lipid metabolism, but when produced in excess, they provide additional energy, reduce satiety hormone like peptide YY and glucagon like peptide 1, and stimulate fat storage in the liver and adipose tissue. This leads to overeating and suppress regulation of adipocyte metabolism, which is fasting-induced adipocyte factor. When beneficial microbes such as Akkermansia muciniphila are depleted, tight function protein weaken and increase the gut permeability, allowing lipopolysaccharide or LPS to leak into the bloodstream. This triggers metabolic endotoxemia, and systemic inflammation also can lead to metabolic disturbance. Leakage of LPS into circulation activate the CD14 receptor through toll-like receptor 4, stimulating the nuclear factor kappa B pathway. This results increase in pro-inflammatory cytokines, creating a state of chronic low-grade inflammation. Over time, this inflammation drives insulin resistance and leptin resistance. The activation of the peroxisome proliferated activated receptor gamma increases due to the elevated level of short-chain fatty acid, which promotes uncontrolled adipogenesis. AMP-activated protein kinase activity also decreases, reducing fatty acid oxidation, which promote lipogenesis and obesity. This imbalance favor fat storage rather than fat burning. Dysfunctional farnesyl S receptor and Takeda G protein coupled receptor 5 signaling will disturb glucose and lipid metabolism. This not only promotes obesity but also raises the risk of related conditions such as type 2 diabetes and cardiovascular disease. The modern tool in microbiome research I have chosen is shotgun metagenomic sequencing, where the DNA is isolated directly from a sample and the entire genome content is randomly fragmented into smaller pieces for sequencing. This provides a much broader picture compared to 16S rRNA sequencing, which only targets a single specific gene region. But what is the purpose of using shotgun metagenomic? First, it allows us to identify the microbiome composition within a community at species and strain level. Second, it can analyze microbiome gene and metabolic pathway, giving insight into the functional potential of the microbiome. Third, it helps identify microbiome biomarker that may be linked to the different disease and also detect a much wider range of microorganisms beyond bacteria. In 2024, there was research that found reduced microbiome diversity in obese individuals. Along with arrangement of Ruminococcus genevus, they also identified virulent factor and antibiotic-resistant genes. This study has shown moderate diagnostic potential with area under the curve of 0.766. In 2023, research revealed the association of gut microbiota disease with geography and ethnicity, and found that Bacteroides, the Thiothiomicron, was positively linked with insulin resistance. To conclude, the gut microbiome plays a crucial role in digestion, metabolism, immunity, and overall health. When the gut microbiome becomes imbalanced, it disrupts dysfunction and contributes significantly to various diseases, including obesity. With modern tools like metagenomic sequencing, fecal microbiota transplantation, and even CRISPR-based intervention, where it not only helps in deeper understanding of how the microbiome influences obesity, but also offer potential targeted treatments in the future. This is the list of references I have used to support my presentation. Thank you for listening.